Good morning. Can you hear me? Can you see my slides?
We can see the slides and we hear you loud and clear.
Great, great. First of all, warm greetings to you all from France. And special thanks to the organizers for inviting me. It's really it's going to be a pleasure to speak at this very first National Cancer Congress Malaysia. And I will talk about the epigenetics and biomarkers for cancer risk. So for those that might not be necessarily experts, I'll give just a short introduction. So the cancer is not only genetic disease, but also epigenetic disease. And we know that at the molecular level, cancer is driven by different genetic changes, but also epigenetic changes. These changes disrupt different processes, such as transcription, DNA repair, checkpoints, and so on. And also, what is now known that these molecular changes are induced largely by environmental factors, by environment in its broadest definition. Therefore, understanding the epigenetic changes, how they disrupt the processes, how they modulate environmental impact, has important bearing on the mechanism, to understand the mechanisms, but also discovering biomarkers and the targets for therapy and prevention. So the epigenetics, or epigenome, is a kind of a software that extends and modulates hardware, genetic code. So that allows actually that in our body, every cell has identical genome, and yet there are so many different cell types and tissues with specialized functions and so on. And this is thanks to the epigenetics that allows certain genes to be on and off, and therefore dictate and maintain, establish and maintain the identity of the cell. This is very important. And of course, in cancer, things go wrong. And that's what we want to understand. So as I said, the epigenetics is interface between the genome and environment. This can be described in very simple model with a pair of socks. So normally when we buy socks, they have the same fabrics, pair of socks, both socks has the same fabrics and the same color, right? And usually we tend to use them and wash them together, right? However, sometimes it can happen that we unpair the socks and then we wash them separately with different clothes. Other clothes might have different colors. And as we do it repeatedly, what we may end up with a different or socks that look different. So in this scenario, the fabrics is genetics. So the material is genetics, and the color is epigenetics. As you can see, epigenetics is much more susceptible to environmental changes, and therefore we can look at the color. We can look at different phenotypes. For example, when we talk about animals or humans, and then based on the different phenotypes, we can link to different epigenome, and then epigenome can actually tell us about the history of exposures, and therefore this may help us also to identify causes. Suspected unknown causes, but also identify markers for cancer risk. In our laboratory, this is a bit busy slide, but captures our experimental approach, where we take advantage of large cohorts or case control studies, population-based cohorts that are coordinated by our epidemiologists at IARC, with a very rich biobank, high-quality biobank samples, so biological samples, but also the associated exposure or clinical data, different models, including cell systems and murine models. We take advantage of cutting-edge technology that allows profiling, epigenomics, and other omics. And also, of course, we identify profiles that might link to the exposure. We interrogate also the data repositories at public domain. Then we replicate the finding, and in specific cases we also do focused mechanistic studies to understand the functional impact of these changes in cellular processes. So most of the studies done in our group, but elsewhere, is on the cancer tissues. And of course, this is important, but I'll tell you that this can also we could have a different approach, which can be also very important, informative. And in this particular example, we study esophageal cancer from high incidence populations. And as you can see here in dark blue, these are the areas where esophageal cancer is one of the major cancer types. While in Europe, for example, it's really rare. And we don't know really the causes, but what we managed to take advantage of the infrastructure established by different colleagues at IARC that collect samples and associated data, exposure data in those regions. And we combine this with powerful epigenomics profile. In this case, we use DNA methylome profiling. And what you can see in the cancer samples in this circus plot that captures differentially methylated positions in a cancer versus control tissue. You can see here hypo in yellow and purple. We see hypermethylated positions. So you can see the epigenome is drastically altered in cancer tissue versus normal non-cancer tissue. So of course, the critical point here is to identify those early events, those driver events, and distinguish them from merely passenger epigenetic events. That's what we are trying to do in most of our studies. But also we want to identify those potentially early markers when we stratify according to the tumor stage, and we identify specific genes, in this case, for example, PAX9, that tend to appear the methylation of which tend to appear early in the tumor, and we try to combine these candidate markers with a minimally invasive biospecimen device, in this case cytosponge. Actually, this is something that we can brush from the esophagus. We can brush the tissues and cells, and then try to see whether the markers, we can identify markers that can be used for risk stratification in low resource settings. So this is ongoing, and we are now testing some of the markers in collaboration with our Chinese colleagues that actually also are interested, because this is also a major problem in some parts of China, and they also have their own cytosponge, and we have a collaborator in our group that now are testing some of those markers that we identified. So we go actually beyond epigenomics and also trying to look at the other layers. So how the epigenome impacts for example, transcriptome. And this is an example that we published recently on cutaneous and acral melanoma that revealed not only the drivers, epigenetic drivers, and also the pathways that are likely to be driving the cancer, but also markers that can be used actually to predict the outcome of the treatment. And this is a very interesting study because we used also genetics to look at the mutations, and that can also tell us about exposures. For example, UV ultraviolet radiation is a known risk factor for cutaneous melanoma, but also can stratify the samples that are linked to the UV versus non-UV. So I think this is an example that we can look at different omics layer and the link epigenome to the other omics layer. So we don't have to actually only look at the tumors, we can look at adjacent normal tissue, and this is a study in collaboration with our Korean colleagues, where the gastric cancer is a major problem and linked to the Helicobacter pylori. So we actually collected mucosa in a normal mucosa, non-tumor mucosa, in cases and controls, and also in those subjects that are Helicobacter pylori positive and the negative. And you can see in the positive, if we profile methylome mucosa, you see the positive cases are very distinct from a negative. And if we eradicate Helicobacter pylori, we see that this mucosa or methylome of the mucosa becomes more like negative. This speaks in favor of the well-known established idea of epigenome being reversible, epigenetic changes being reversible compared to genetic changes. So one interesting area or more recent area of research in our lab is to identify the epigenetic changes that are linked to the early life or in utero early life exposure and cancer risk in childhood. Why this is important? Because the epigenome undergo dramatic reprogramming during in utero life, and there are windows of susceptibility that may impact the epigenome. Different exposures during in utero life may impact the epigenome, and these changes can be propagated and constitute a risk in childhood and even later life. And this is the concept, as you may know, this is the developmental origin of health and disease in later life, so-called DOHA. So for this, we took advantage of put together different cohort into a consortium. That this is a mother-child cohorts with the samples taken at birth, but also the rich exposure information during utero life. And so we profiled the epigenome at birth, and then we looked at how they relate to the either exposure during utero life or risk of childhood cancer. So we have an example. This is an example, one of the most striking examples, how the in utero exposure can leave the signature in samples at birth, in this case, so-called cord blood samples. And so we put together different cohorts from 3 different consortia, mounting to more than 6,000 samples analyzed. And then when we looked, for example, how the methylome profile relate to the smoking during pregnancy, and you can see in this Manhattan plot top hits that link the smoking during pregnancy and methylome changes in the cord blood of the baby. And then just the one of more striking example is this AHRR top hit. This is aryl hydrocarbon receptor repressor that is actually, you can see the p-value. So with that almost 10 to the minus 200. So with that confidence, you can say that the methylation of this gene is linking the baby's blood to the mother and to the smoking during pregnancy. So this just illustrates an example of how the exposure can leave a robust signature that can be detected in the blood of the baby. So we also looked the different signature of intermediate phenotype, for example, birth weight, but also can we identify something that can predict the risk. And indeed, we have a limited number of CPG, so-called differentially methylated CPG, that are mapping to specific genes and that predict the risk of leukemia. And they are shown here. We are actually submitting a paper that we characterized. What does this actually mean? And we replicate the finding, but also perform longitudinal analysis in samples at diagnosis and also in post-diagnosis samples. So basically something that we can see at birth, and then we can see how the stability of these markers at diagnosis, and then whether also these markers are predicting the remission or outcome of the treatment. So just a couple of minutes to talk about the other markers, but this here is the markers in adult cancer, specifically in breast cancer, and it was a pleasure to see that our former postdoc, now independent researcher in Stanford University, who is chairing the session, Felicia Chang, who actually spent a few years in our lab, and she actually contributed to several projects, and one of those actually I will present here in next couple of slides. And this is a hypothetical model that we can actually look not only in tumors, normal adjacent tissue, but also in so-called surrogate tissue, where we can identify markers that can actually help us to stratify, predict the risk, or stratify risk of cancer in different types of cancer. So this is based on the concept that different environmental exposure or stochastic events that may occur early in utero life or even in later life, but in the stem or progenitor cell. As you know, each tissue has its own adult specific stem cells, and therefore these changes in the epigenome can be stably propagated and can create mosaicism, or can be more kind of spread around the bodies, and therefore we could study in surrogate tissue, for example, in blood, that is often accessible, more accessible than other tissues, and to see whether we can identify markers of risk. We were encouraged by our study on the epigenetic clock in a prospective study, in European prospective study, so-called EPIC, where we profile methylome and look at this algorithm for epigenetic clock. And then we could see that for example, in breast cancer, in prospectively collected samples of future breast cancer cases, epigenetic clock is accelerated. So what Felicia did actually in this study is to look at the prospective samples that are collected at different times prior to diagnosis of breast cancer. So this study is taking advantage of the EPIC cohort, so the cohort that were large prospective cohort in Europe, and then also this technology called RRBS, Reduced Representation Biopsy File Sequencing, that is having a good coverage and is based on the deep sequencing, next generation sequencing. So without going too much details, but Felicia really took a leadership in analyzing and interpreting these data sets that are not trivial compared to the others, for example, for which we have a robust pipelines. And so she basically ended up to identify using the machine learning and so on, to identify sets of differentially methylated positions in the blood that can predict breast cancer up to several years, even 15 years prior to diagnosis. Obviously, these are not diagnostic markers, but these are the markers of risk, so they can be used to stratify and included in different models, for example, in conjunction with the screening, it predict the risk of in that individual at high risk of developing breast cancer. So we also argue that the epigenetic should be incorporated in a so-called personalized early detection and prevention of breast cancer, and this actually specifically might be important where we want to de-escalate the screening, which we know has its own disadvantages, and we have this position paper published in Nature Review Clinical Oncology. So obviously we want to argue that this type of markers, epigenetic, new generation epigenetic markers, can be incorporated for other cancer types. So we also looked whether because epigenetic changes are in principle reversible, we want to see whether how the different risk conditions, for example, metabolic syndrome, obesity, whether they can contribute to whether the epigenetic changes contribute, it has a causal role, or also can be modulated by changing the lifestyle. For example, in this particular case, whether the weight loss can modulate the epigenetic changes and then reduce the risk of colorectal cancer. This is a relatively early stage, but we were able to demonstrate that the people that are obese, when they lose the weight, either through the caloric restriction or metformin treatment and so on, either healthy people, so a non-cancer patient or survivors, that there is a positive impact on the epigenetic changes and reduction, so-called epigenetic aging or epigenetic clock is slowed down or reversed. So this, we have published this last year, and we continue to see how this impacts the cancer risk. So the last slide that I want to show you is that the epigenetic changes may not necessarily always be bad thing or causally involved cancer, but also in this particular study we showed that the certain lifestyles, for example, coffee or tea drinking, might have actually positive impact on epigenetic changes. And we characterize this and point down to some of the liver metabolism pathways that might be modulated, and how the coffee, for example, drinking might have a positive impact on health. I will skip, we have one interesting line, but in the interest of time I will skip it. How to functionally characterize driver events in cancer, and I'll just skip this, and I think in the interest of time I'll just conclude. The epigenome now considered as a mainstream, or epigenetics is considered the mainstream cancer research, is an interface between the genome and the environment. And I will give you, I have given you some examples that the epigenome is profoundly deregulated in cancer, and that also looking at the epigenetic profiles can identify fingerprints or signatures of exposure, but also markers of risk or clinical outcome. Also, there was an example of how we can apply this cutting-edge technology to population-based cohorts and biobanks in order to gain insight into the risk factors and identify biomarkers. I think in the future work we are aimed at understanding the role, the regulation in causal pathways in different cancer types and different suspected, known or suspected risk factors, but also to provide new biomarkers to identify and validate biomarkers that can be used for early detection of cancer risk stratification, where actually this could be particularly useful for so-called implementation research. So this actually is captured here, so a different across the cancer continuum. So the epigenetic characterizing epigenome can provide the markers for risk, but also markers of diagnosis, prognosis, as well as the targets for personalized therapy and prevention. So I'd like to acknowledge many collaborators, including some in Malaysia, and Felicia Chang is one of those. It was really a privilege to had a privilege to visit. Could not be today with you, but it was a pleasure a couple of years ago that I visited your country, and I hope that there will be another opportunity soon. And last but not least, my team at IARC. These are the people that actually are behind the work that I present. So I'm really privileged to lead this nice and enthusiastic group. Thank you very much.
Thank you very much, Zdenko. That was a very nice talk, nicely encapsulating a wide range of studies. So now we have the Q&A session for both Dr. Zdenko and Dr. Ho. We're very interesting about epigenetic biomarkers of risk, and one is genetic biomarkers of polygenic risk scores. So could we have a few minutes for just maybe 3 or 4 questions? So may I ask the audience, does anyone have any questions for? Yes. Maybe you can say it and I'll repeat it for the benefit of those online.
Alright, I'm a master's student currently doing research on epigenetics. So my question is, when I talk about, like, for my understanding, I know methylation and hypermethylation, but unmethylated and methylated genes, like there is hypermethylation and there is unmethylated. And when there is one partial methylation, I want to know the cause of why partial methylation occurs and how it affects the body.
Right, so this I presume is directed to Zdenko. So the question from a student here who is working with DNA methylation. She often sees
Yes.
hypermethylation and
Very,
hypomethylation.
very nice, very important question.
Yeah,
So,
did you hear it?
so the,
Okay,
so,
okay, go ahead then.
oh, sorry,
No, I assumed you couldn't hear it, but if you could hear it, yes, go ahead and answer, please.
so shall I answer or?
Yes,
you
no,
answer?
you answer.
Yes, so the for mutations, it's very clear when it's off and on. You know, you mutate the gene and the gene is off. Now for methylation, so you have in a gene promoter, for example, you have a number of CPG sites. So there is not only one CPG site. Often there is a number of CPG sites, and they tend to be they may be unmethylated. All of them can be all of them methylated, polymethylated, or can be partially. So it means in the single cell, if you look at the single cell level, so the methylation can be the CPG can be methylated or unmethylated. But if you look the region, promoter region, some CPGs can be methylated, unmethylated. So when you talk about partial methylation, so this means that some genes, some CPGs are methylated, some are not, or you have actually when you analyze the sample, it's a mixture of cells. So you are, you get average of methylation. So that's why it can be 50% or 70% actually, and so that's why it looks like partial methylation. So usually when gene is fully methylated, it's associated with the silencing, and unmethylated with the active genes. Although this is not always the case. We know that the genes that might be unmethylated, but they are not expressed through other mechanisms. For example, you need machinery to allow expression. So even if this gene is unmethylated, it's not always expressed. I don't know whether I answered. There are two things. So there are several CPGs, and also we analyze the mixture of cells, and therefore we are averaging. So if you look at the single cell, of course, there will be less of a problem in interpreting.
Okay, thank you. Are there any other questions? Yes, go for it.
Okay, I'm Dana, dental postgraduate student. Okay, currently I'm actually doing research on public health aspects of screening or cancer screening. But just to come into the context, I would ask about the cancer risk assessment. This one, is it have been used anywhere, not on Indonesia or anywhere, because as we know, mass population screening is very expensive. So is it has been applied anywhere to use this cancer risk assessment for their targeted screening?
Right. So maybe Dr. Ho can take this.
So your question is, has cancer risk assessment tool been implemented in anywhere in Malaysia?
For targeted screening, yeah, Malaysia or
In the West, so including genetic in cancer risk assessment. So there's a few clinical trial that's going on in the UK and in the US, where they want to, they're using a cancer risk assessment model that look into lifestyle, mammogram images, as well as genetic, but they are not rolling it out yet. So at the moment, they are trying to identify if they offer this to women, then how can they incorporate that into their population-based screening. So you make a really good point, because genetic testing is expensive. So the questions that I get almost at every single talk is, how could that be useful in the context of our country? My answer to that today, slightly different from last time, would be you always need to have it. So it's like building it's like a Bentley. So I have a fancy model that incorporates everything, but can people afford it is then a practical issue. That's why what our team is now working on, because we need to talk about genetic, that's why I didn't raise that out, is can you actually extract important information from the mammogram itself that can offer the same predictive accuracy as the genetic. So best is I have everything. But if I cannot have everything, maybe I can rely the information from the mammogram to do the risk stratified screening. Yeah.
Okay, I
see some more hands, but I'm sorry that the organizers are staring at me. We need to end this session. Okay? So Dr. Ho will be here with us. Well, I'm not sure for the whole 3 days or not, but she's in Nottingham, Malaysia. You can catch her. And Zdenko, I can attest that he is super approachable and very, very helpful and friendly. So with that, I will have to end the session. Thank you very much to our speakers, including Zdenko. I will now pass the time back to the organizers who will make the announcements.